Well, thank you so much. It's great to be here. My name is Orlando, and I'm from North Carolina State University. My collaborator, Kevin Flores, and I are working on this problem, which is how the mitochondria is inherited through sex cell reproduction. And the mitochondria in this cartoon are the green dots, and as you can see, it basically changes a lot during this process. And we know a lot about how other molecules and structures like the DNA are inherited through sex cell production. But we actually know surprisingly little about how the mitochondria is inherited. And the game here is basically the nuclei are going to be split into four. So we start with one gray dot, and then we end up with four gray dots. And the mitochondria somehow has to be deposited in each sex cell, and we don't know exactly how that happens. When the cells fail at this process, you might have problems like progeny that is infertile, and it can also lead to cardiomyopathies and muscular dystrophy in humans. The problem here is not only how the mitochondria gets to the sex cells, but also the fact that that process is multidimensional. So we have a lot of different proteins and networks being involved into this process, and we don't know exactly how those signaling proteins are connected. So to visualize all this multidimensional complexity, we created a microscopy system that allows us to track single cells. In this case, these are G cells, and we can look at up to 6 different fluorescent markers in these cells and obtain long-term time series that tell us how the system evolves through the process of cell division and sexual reproduction. And our hope is to basically take those time series and use biologically informed neural networks to create a minimalist model that will be potentially the first comprehensive mathematical model that explains how the mitochondria is inherited in sex cells. Thank you so much.
Thanks for the opportunity to, let me see. Okay, great. Thanks for the opportunity. So I'm Ratul Biswas. I'm at the University of Minnesota pursuing my PhD, and I'll be joining NITMB in the fall as a postdoctoral scholar. Today I'll be talking about dilute spin glasses and how there are some biological applications. So let me just give you a quick one-line intro to spin glasses. So spin glasses are models in statistical physics which came up when people were trying to understand magnetic properties of metal alloys, and later people realized that there are some complexities in biological systems which have same features as spin glasses. So some of the biological systems that come under the umbrella of spin glass theory is the first one is neural networks. So we are trying to understand the function of the brain in absorbing and retaining new information. People are also interested in protein folding, how the primary structure folds through secondary tertiary structures to give you the native functional form. And there is also application of spin glasses to community detection, where people are interested in studying predator-prey models and modeling interactions between different populations in the society. So here's my research. So through my graduate time, I have been able to provide a general framework to study dilute models rigorously from a mathematical perspective. We identify parameter regimes where models behave nicely, and for those who are experts, I mean replica symmetric behavior. And we have been able to validate old formulas which are true for discrete models, and we also derive new formulas which are true for continuous state models. And future directions include studying models which are increasingly more complex. So with that, I'll thank you for your attention.
Right. I'm Andreas Puttenschön. I'm a newish assistant professor at the University of Massachusetts Amherst, and I'm an applied mathematician whose work is really motivated by consensus formation and cell migration in tissue systems. From simple systems up there with a few cells to complicated systems with many cells. Now, I use a stepwise mathematical modeling approach where I start with constructing really detailed models that include a lot of details. So for instance, here we have a CPM cell with some small GTPase proteins that control the cytoskeleton. A little bit further up, we have a physics-inspired model where we can describe things like nuclei, filopodia, and how how the cells interact with this meshwork of mechanical extracellular matrix around this. The challenge is that these complicated models are really hard to analyze and to extract fundamental principles from. So what I really like to do is I like to coarse-grain them to non-local ODE models, which improve the situation somewhat, but not too much. And I really like to go to mean field limits, where I was trained to do bifurcation analysis and dynamical systems analysis of these types of models. And so what I've been recently gotten really excited about is that a lot of the insights of bifurcation analysis in this mean field setting translate back to allow us to make predictions for very, very relatively small cell groups of the size of 10, 20, and we can really nicely understand how to pick physically realistic parameters in these types of complicated systems. Thank you.
Hi, I'm Dan Cooney, newish assistant professor at University of Illinois Urbana-Champaign and new affiliate member of NITMB. And I'm here I want to talk about one of my favorite questions in evolutionary biology, thinking about sort of the evolutionary dynamics across different levels of selection, and these can arise in a range of situations from sort of origins of life and the origins of chromosomes to human cultural evolution. And a lot of these problems are summarized by some tension between individual level incentives to cheat and some collective incentive to cooperate. And so I like starting, as Andreas did, with agent-based or individual-based models and derive mean field limits here, for example, talking about the collection of groups with certain levels of cooperative individuals due to both the effects of within-group and between-group competition and understanding when sort of collectively beneficial things can arise. And there are really lots of fun behaviors you can see in these models, such as the level of group-level competition required to sustain cooperative behavior arising as a tug-of-war between these individual and group-level incentives, and some really fun effects where, for example, when groups are most favored by cooperation, you can get as much cooperation as possible by incorporating cooperation between group, and some fun behaviors where, say, 75% groups with a mixed level of cooperation are most favored under group level competition, but yet you see less cooperation that's optimal, even in limits of strong competition, an effect I used to like to call the ghost of lower level selection. So I'd be happy to talk about all these cool models of cross-scale evolutionary dynamics in some of the breaks if anyone's interested in learning more. And it was really fun. Starting these sorts of projects actually arose from a conversation with NITMB member Carl Veller from U Chicago over a lunch. So I guess I owe him the next time we have a lunch as well.
I'm Bob Eisenberg, and I'm talking to you about mitochondria. Mitochondria are, in a way, the most important single organelle in life because they generate ATP, which in the enormous diversity of life is almost the only energy currency used anywhere. There's been a huge amount of work done over the years on doing something wrong with the point here. Anyway, on mitochondria, and there is an enormous amount of molecular detail in the hundreds or even thousands of papers done on this. But there is not a model that predicts how ATP is actually produced. And in fact, believe it or not, and you can check this yourself, in the hundreds and hundreds of papers discussing electron transport, their word electric current does not appear. That suggests that one of the difficulties in making these models is perhaps that they didn't know about electricity. Could we advance? There we go. So, looking at this, what are we going to say about electricity? Well, we're going to start with the Ampere-Maxwell law, the second equation there. And I want to point out to you that this is a theory of everything electrical. There are no known errors in this equation. It works inside hydrogen bombs. It's used to design hydrogen bombs, and it works on all other scales. So now we simply take the divergence of that equation, and we look at the blue equation. Div curl is 0 by an identity. That tells us something remarkable. It says that the quantity I'm calling total current is 0. That has 0 divergence and is perfectly conserved. That is the current law that is needed to understand mitochondria.
All right. Hello, everyone. I'm Federica Ferretti. I'm one of the NITMB fellows. One of the peculiar challenges of evolution is that it has to balance these two things, efficiency of natural selection on one hand, and robustness to changes in the selection conditions. And one example of a biological situation where this tension is at play is in adaptive immunity, where, as you know, we produce antibodies in response to an infection, to a new pathogen, but what we really want is not something which is overfit for the pathogen that we've seen, but something that is potentially also able to neutralize mutants of that pathogen. And so this tension is of the same kind that is at play in machine learning, where you try to balance between optimization and generalization. And so motivated by that analogy and motivated by the biological insight that comes from affinity maturation, we decided to study a simple model, population dynamics model, where we introduce individual mini-batching, meaning that each individual in the population is allowed to make an independent choice of the environmental conditions that they live in at each generation. And so we're able to characterize analytically the noise that comes from these stochastic choices and to show that if certain conditions are satisfied, we are actually gaining in generalization. Sorry, my bad. We're actually gaining in generalization abilities, and this happens because the interplay between the nonlinearities of the systems and of the system and the noise creates a non-equilibrium trap that traps the population away from where it would be. For more details, there is a poster. Thank you.
Okay, I'm going to tell you about some hibernation stuff today. Okay, so squirrels running around northern Alaska. Its body temperature does this. So this is like while it's hibernating, you get these bifurcations. And people don't know what drives the spiking behavior, right? So at some point, right, like you kind of get stuck biologically. One group thinks that the thing driving it has this kind of behavior, so you have a non-fixed period. Here you have a fixed period, right? Okay, why do people care about these things, right? Understanding their physiology, we can maybe, it's thought that, I don't know what to say exactly, but basically like key to long-distance space travel is thought like maybe this could be beneficial or extending our physiology. Let me show here. So what do? We do a bunch of mathematical modeling, hardcore model selection, supercomputing, but basically we infer the hidden state and we show that it must have a fixed period. And so using that, we can differentiate between kind of theorized mechanisms. One last point, if I go back, all kinds of different organisms do this, right? Bears, shrews, birds, and we show that using the mathematics, we can show that there may exist universal thermoregulatory mechanism. And that's all.
Hello, everybody. I'm Ben Kuznets-Beck, and today I'm going to be telling you guys about transcriptome-wide linear response. So you may not be surprised to know that if you change the rate of transcription of a gene, then its number of transcripts will change. But you might be a little bit more surprised to know that there's a cascading effect due to the fact that genes live within the context of a gene regulatory network. So the point being that linear response basically relates the perturbed response of some dynamical system to how it fluctuates in the absence of that perturbation. And so if we're looking at gene expression changes, then the expression change of gene I is just going to be the sum over all j of the fluctuations, how it correlates with gene J times the perturbation U. And so we can basically solve for what perturbations we need in order for linear response to work by just inverting this correlation matrix sigma. And so we tried this on essentially a toy model of genes competing on teams. So we have 10 genes on each team. They're mutually activating within the team and mutually inhibiting across teams. And we basically allow for them to burst with these Hill function rates. And we find that, of course, linear response does indeed work here. We see that we get nice block correlations, and that if we were to try to predict the change in expression, we do that very well with an R squared close to 1. If we look at actual data now, we can look at, for example, gene knockdown. So this is the log fold change for all the genes. We see ACT B as the one that's actually knocked down. If we were to try to predict which gene was knocked down, we would see indeed it was ACT B. And we see here that the expression change across the whole genome-wide profile is well recapitulated by linear response. We also see that if we just push on a single gene, that we can do pretty well. So for example, we know that ARID1A here was knocked down. We push on that single gene and we see an R squared of around 0.4 here. So again, the predicted response from linear response is in blue, and the orange shows the observed response. Again, we can push on MAPK3 too, and we see an R squared of around 0.67. So linear response does a good job of recapitulating how genes change their transcription in a transcriptome-wide way.
Yeah, soils are everywhere. Maybe not so much in New York, but it's doing a lot of functions. It's doing some nitrogen cycling, carbon cycling with the microbes, and many people who are studying soils gave up explaining the complex collective behavior because it's so complex. Yet we did not give up, and we approached this problem with two simple strategies, with a philosophy that if we perturb the system and then we measure carefully the dynamics, we would be able to explain this. So I go out into the field and I sample from different native pH and then perturb the pH in the lab. And then I carefully measure the nitrate, nitrite, different metabolites that are involved in nitrogen cycling. These are the dynamics. And the model surprisingly captures all of this behavior with one single biomass field. And Ziqi will talk a little bit more, who is our co-first author. And so this is the flagship paper that we have, a revision in Nature. And with Matt and Vincenzo's group, we now want to expand this into, we only use one biomass field, but what if we can have multi-biomass? So we want to coarse grain these thousands and thousands of species so that it can explain the function better. We have 10 terabytes of metagenomics data that we got. And now we want to also ask, now not only taxa, but can we use genes to explain this function better? And so now we are working on this. If you need some data, come to me, it's free.
Okay, I'll talk about the theory part of this project. So we want to learn how the soil microbe community responds to the environmental change. So here, the environment means the pH perturbation. And the response is the metabolite dynamics. So here, we measure the nutrient dynamics. So the puzzle here is that we have, in the experiments, we have two conditions. In the control conditions, we have some drugs. So there is no growth of the cell. So you can imagine that the cell can eat the nutrient with constant rate. So you can see the red line is almost linear, right? And if there is no drug, the cell can grow. So they can grow, they can eat the nutrient faster and faster. So you wish to see some nonlinear dynamics of the nitrate. However, in experiment, you see the linear nitrate. So it's very weird. It seems that the cells can grow, but it can grow a bit, and then they stop growing, and the rate is constant. So what happens? So in the model, we imagine that we hypothesis that there are some limited hidden nutrient there. It's very small. So when they run out of this hidden nutrient, they stop growing. So we see the linear thing. So we use the consumer resource model. So in this system, when the hidden nutrient and the nutrient we put in are both larger than 0, they can grow. But if one of them is close to 0, they stop growing. So that's a model. And we tested the model. We fit the data with the model, and we found out that when there is high pH, there is more hidden nutrient. And also, we do some other experiments. We arbitrarily add some hidden nutrient to the system, and we see that the system actually as predicted. So there are some carbons in the soil that is a hidden nutrient, and we perturb the pH, we perturb the carbon. So this will change the microbe community. Thank you.
Let's keep the bacteria theme going. Let's talk about oscillations. I am Yu Jia, a grad student working with Mike Rust at the University of Chicago. So here, circadian rhythm, or the day-night rhythm, found in many branches of the tree of life, is driven by a biochemical oscillator. Quite remarkably, basically all of these circadian oscillators share the same feature, which is that their period is not sensitive to the change in temperature, known as temperature compensation. Nonetheless, temperature goes into this picture by acting as an input signal to tune the amplitude. Here, cyanobacteria, they have one of the simplest circadian oscillator systems. With them, we have this method to now measure not only the amplitude and period of the circadian oscillator of cyanobacteria, but how the attractor moves and deforms in this phosphorylation phase space. And this opens doors to many questions. For example, we ask how temperature combines with other input signals, such as light. With more measurements of the biochemical processes and the ODE model, we also ask what is the molecular basis of temperature compensation. So our working model here is that there is a balance between changes in kinetics and feedback. The former tunes the speed of the system moving around the orbit, and the latter tunes the size of the orbit. We'll have to see how real this picture is. The work, we cannot achieve any of these without Gopal and our collaborator Chris from Aaron Dinner's lab and the funding from NITMB. Thank you.
Okay. Now for something a little bit different. My name is Julius Lux. I'm going to tell you about some work that I'm doing with Rishi Kondor through the NITMB. We're really interested in solving the dynamic RNA folding problem. So why RNA? Well, I think RNA is life's central molecule. It's the only molecule we know of that can store and propagate genetic information as well as perform functions critical to life. The way that it does that is by folding into very intricate shapes, and that folding process is highly dynamic and complex. If you imagine molecules folding on some sort of free energy landscape, that landscape is in fact changing as each nucleotide of the RNA is polymerized, as you can kind of see from some of these reconstructions here. Actually, very similar to some of the things that Eric talked about today, we're interested about broad properties of these energy landscapes. And in fact, whether or not natural RNA sequences somehow encode landscape changes that allow very efficient folds. There's a lot of propensity for RNA to get trapped into kinetic traps, and we're wondering if these landscape encode things like folding funnels and so on. With Rishi, we're taking a little bit of a different approach and asking, can we skip the structure piece and go straight to developing models that can predict RNA function from sequence alone, using some of the mathematical properties of graph neural networks that can actually encode structural information inside the network itself. Finally, we do this not only to understand fundamental RNAs in nature, but also to design RNA biotechnology. So half of my lab actually focuses on developing some of these tools. On the bottom, shown some data of an RNA biosensor that can detect if water is contaminated with fluoride or not, and these are some results from a field trial we just completed in Kenya. So thank you.
Hi everyone, I'm Sergey Maslov, an oldish professor from UIUC and a new faculty affiliate of NITMB. I'll give you kind of a little taste of the work we have been doing with my current student Zihan Wang and my former student collaborators Jacopo Grilli and Akshit Goyal. And it's about microbial ecology, so we are back to microbes. So the microbes actually different microbes use different metabolic strategies to consume resources. Some microbes are a little bit like cookie monsters. When you give them a mixture of resources, they eat all of them at once and deplete them pretty much at the same time. Other microbes like E. coli, for instance, presented with a variety of sugars, eat them one at a time. They are like a typical kid who will first eat sweets and then eat maybe a burger and then finish with broccoli. And finally, some microbes are really like picky eaters. They only eat one resource and don't touch anything else. So the question is, we don't really understand how the ecological environments in which microbes coexist shapes which of those three metabolic strategies is optimal. In fact, a recent review told that we know a lot about how these shifts in, for instance, hierarchical utilization here are regulated. We even have a 500 parameter model for how they are regulated in E. coli. We know a little bit about physiology, how these particular strategies are optimal while microbes are grown alone. And one of the key parameters here is how much proteome is pre-allocated to the resource you are not currently using. And finally, we know next to nothing about what is ecological context favoring strategies over one strategy over the other. So in our recent series of papers, we kind of try to address it. And this is, I'm presenting here an unpublished work. Basically, the idea is that if you will vary the fluctuation, the strength of the fluctuation of the resources, you will see a systematic change of the strategy between specialists, which dominate for very stable environments. Dioxers will dominate for weakly fluctuating environment, and co-utilizers will dominate for strongly fluctuating environments. The other parameter of this diagram is how equal are resources to each other. And what is encouraging is that this optimal, it's an evolutionary model which we evolve in silico, and this parameter for pre-allocation of resources towards unused nutrients is reasonable. It's about a few percent, which is what experimentally we see in many microbes. Again, if you want to know more, come to my poster. Thank you.
Hi everyone, my name is Madeline and I'm a PhD student in Yogesh Goyal's lab at Northwestern. And today I'm going to talk to you about how we're tracking cell adaptation to perturbations. And this is a collaboration with Arvind Murugan and Kabir Hussain, Arvind Murugan at UChicago. And so people have been very interested in finding out how things adapt to perturbations using just endpoint measurements for a long time, so all the way back to the Luria-Delbruck experiment, which was Nobel Prize winning. So they determined that bacterial resistance to phage was as a result of spontaneous, so pre-existing mutations rather than induced mutations. And they did this with just endpoint measurements of the number of resistant clones and statistical inference alone. But when you try to apply those same principles to mammalian resistance, so for example, a cancer cell becoming resistant to an anti-cancer drug, you can't really do it because the mammalian cell resistance is a lot more complex, largely due, or in a large part due to component of non-genetic resistance, which is just the cell transcriptome, so all the RNA, not the DNA changing at all, and can have a lot of dynamic nuances. And people have tried to mathematically model this in the past, but this is still not capturing the non-genetic component in as much as it does have an influence. And it also, you're getting disjointed snapshots, so you're not able to sample the same cell clone over time. And so what we want to do is kind of connect the dots between the initial clones and the final clones and track how the clones change over time. And how we're going to do this is using a recently developed system by Michael Elowitz's lab, where you can target a specific mRNA for export by the cell and then sample the media, sequence the media, and determine how much of each RNA is inside the population. And instead of a specific gene, we are going to label DNA barcodes, which we can add to the cells. It labels a specific clone uniquely, and then so we can sample the media and collect information about all the clone sizes of all the barcodes, all the cells inside that are adapting at a specific time. And this can be very useful for identifying not only the drug treatment experiments, but also other contexts such as CRISPR screens.
As you know, protein structure can be it can be given by a sequence of amino acids, and the structure and wild-type protein, it has its own function. Similar to that, we consider a network with a sequence of conductances. That the task of that network could be that we apply voltages on input nodes and measure voltages at output nodes, and we can train this resistor network for a desired task. So we analyze, after training this network, we can mutate on the conductance of edges. And if we have some non-additive effect of mutation on the function of the network, like a protein, we say that we have epistasis. And when there is a nonlinear function of an additive trait in our system, we say that we like proteins. We say that we have global epistasis. Interestingly, we see that there is a global epistasis behavior in our system. We analyze this system, the network system, in the framework of the cost Hessian and loss Hessian. And we calculate the eigenvalues of that trained network. And we see that in the trained network, there's a big gap in the eigenvalue spectrum of our system, which suggested us that maybe proteins are overparameterized with a few large eigenvalues, or which means that if we mutate our system in those special directions, we will see a large changes in its function. And if you are interested in this work, I would be happy to discuss about it in the break time. Thank you.
Great. I'm Sean. I'm a postdoc at Emory. I'm currently funded by one of the external grants from the Institute. You need to turn on the clock or I'm never going to stop talking. And I'm going to tell you a bit about a couple directions we're going, trying to understand something about glucose regulation and diabetes with math and theory. So in healthy people, the concentration of glucose in the blood is regulated. And the main way that this happens is through secretion of a hormone called insulin, which is secreted when blood glucose is high and lowers the blood glucose. Diabetes is a disease where this regulation fails, where the blood glucose is high and it has very slow dynamics after a meal. And our goal is to sort of use mathematical theory to understand both the regulation, the control in the healthy body, and also the disease where it breaks down. Okay, and so to this end, we're working in a couple different directions. One of the areas we're working is trying to develop models which describe the dynamics of specific subtypes of diabetes, because it turns out that this disease manifests in very different ways in different people, and we have sort of no even coarse-grained understanding of how these different kinds of dynamics can appear. So in this case, we've developed a coarse-grained model for a specific subtype of diabetes, which has very unusual dynamics. In this subtype, the disease develops much, much more rapidly than it does in typical patients. People show up in the hospital, in the emergency room, basically dying, and they claim that nothing was wrong with them 3 weeks ago. And then on the other side of it, they can get better after months of treatment and then apparently go back to normal, not needing any further treatment. And we've sort of been able to figure out how to explain this dramatic difference by postulating the existence of sort of an unknown process that exists in these patients, but not in other patients. So I have a poster about this, and we have a paper, but you can come to my poster after we're done here and talk about it. I hate using this. For example, there are 3 or 4 other hormones that interact with the hormone insulin, and really there's no explanation at all of why they have these interactions the way they do. And in this direction, we sort of have developed a hypothesis that the reason that there are all these interactions that no one knows the purpose of is that in reality, for most of human evolution, there have been things that are much more important than being able to pound 5 Cokes and rapidly digest them. The system has evolved to achieve many more other important metabolic goals, not just prevent diabetes. And the hope is that by sort of developing a control theory of how to achieve all these different goals together, we can sort of understand the dynamics of these other components of the network. And I don't have a poster about that, but I'd be happy to draw on the back of a napkin if you catch me in the break. So thank you.
Hello, I'm Thomas Whitehawk from Northwestern University. My collaborators are listed down here, and if you want to get tossed out of the room, you can snap the QR code to see the paper. So I'm going to talk about irreversibility in gene regulatory networks, and I'm going to start with the fact that everyone in this room probably appreciates is that DNA contains the information of the cell, and the proteins encode the phenotype. And if you take this to the extreme, you'd say that if you give me a DNA sequence, there's a unique phenotype associated with that. Of course, our bodies are a counterexample because your cells do a whole bunch of different functions with the same DNA. So in this paper, we're asking about whether this can be done in E. coli. They don't have the same epigenetic mechanisms that we do. And specifically, we're asking whether you can transiently turn a gene off and then turn it back on and leave the cell in a new state. And our strategy is to use the reconstructed regulatory network of E. coli, assign Boolean dynamics to it that's consistent with that network structure, and then map out the attractors of that structure. On top of these stable states, we will apply a single gene perturbation, let the system relax, remove the perturbation, and then see whether the genes are in a new state, which is what's encoded in red. And then in the similar spirit to earlier talks, we're trying to average over models to take into account the fact we don't know how all of these rules are going to interact with each other, so we generate an ensemble of rules that are consistent with the network structure. Here is our key result. We're showing the average irreversibility of the genes, and in the background you can see the strongly connected components of the network are highlighted in that light purple. And we see that each of these strongly connected components has positive circuits in it, which is a necessary aspect of irreversibility. It connects the structure to the irreversibility we observe. Next, depending on how important or the ordering of the genes, we get very different profiles of whether genes admit irreversible perturbations. Because we're using a whole network formalism, we can connect the gene responses to existing data, at least at the level of the initial perturbation. And this motivated us to condense down to a simple phenomenological model for the key motif, which is a 2-gene motif. And we analyze that with differential equations to find phenomenological parameters associated with irreversibility. Thank you very much.
Hi, I'm Maria. I'm a graduate student with Pankaj Mehta at Boston University. Very broadly speaking, what I work on are cell-fate transitions, and we heard a bit about that from Eric this morning. But the very broad biological overview is that complex animals like humans and mice have all of these very highly specialized cell types that work together. And the question is, how do you get from a single cell to this very highly complex organism? And how do you develop all of these cell fates, and how do they transition? And can we find some general principles like universal classes that underlie these cell fate transitions? So, a few key points throughout my work are basically, building off of Eric's work on formalizing the Waddington landscape. Basically, you can model these high-dimensional gene regulatory networks as low-dimensional landscapes. You can use some theory and some mathematical tools from Hopfield models to define the coordinates of sulfate space and look at gene expression data on these coordinates. And you can also combine gradient-like dynamical systems with Hopfield models to basically identify the signatures of these different classes, which correspond to different topological and different broad qualitative behaviors, as well as different landscapes. Thank you.